Rachel Finck is the Senior Director of Informatics at Ionpath. She leads the software and bioinformatics efforts, and this team where they use multiplex ion beam imaging platform as well. Rachel has been working with MIBI data since the time of its conception, when she was working in the Gary Nolan lab at Stanford. And she co-authored the first publication identifying this technique. There she also developed other methods of processing and analyzing mass cytometry data. And we're very privileged to have her here to speak with us, so please help me welcome Rachel to the podium.
So thanks a lot for having me today. Today I have the opportunity to speak after Liat, who's talked so beautifully about the new technology and a particular application of it, that I'm going to talk about a completely different aspect of working with multiplex ion beam imaging data. And what I'm going to focus on is data management and visualization through a web application. And although I'm going to do that in the context of MIBI, and in particular with an application that we've developed at Ionpath, I think that many of you will find the challenges that motivated this familiar from any of the data types that all of you work with, and I hope that as well you'll find the approach that we've taken to address them relevant. And so when I talk about data management and visualization, where am I coming from? Well, early on at Ionpath, we started developing this for our own internal use. It was very selfish on our part because having worked with MIBI and scTECH and other data types before, I knew firsthand the balance of time you spend on finding the files of interest, renaming things, just making some plots for sanity checks, as compared to actually doing data analysis. And we want people internally, and we want users to spend more time on the interesting parts of data analysis, which is largely what we've heard about today, rather than data munging. And so in particular, what are the types of features that I'm talking about? What I'm talking about is, quick browsing of the data from, something being created to be able to just look at it, get some intuition, do those sanity checks. That's different from the types of plots that Liat was showing towards the end there with all these beautiful results, but it's a way to get there. It's a way to get there faster. Also talking about being able to search your data. It's really, really powerful to be able to say, find me all of the images I've ever collected that have CD4 and CD8 and also PDL1, and maybe you're on these tissue types but not others. And so if you can kind of start that practice at the very beginning, it's hugely enabling for all your work later on. You also want to think about how do you get data into the system, how do you get data out of it, what format do I use, all the unglamorous things that have a big impact on efficiency. And very much kind of, Miriam said it better than I could have in her introduction today, but that collaboration, there's sharing of your data once it's published, but there's also this interplay and interaction between there's a mathematician or bioinformatician or software person, working on data processing analysis. I work very closely with pathologists, and being able to just back and forth do something and let her look at it, give quick feedback, know that we're all looking at the same thing, and have the questions that we're asking be the right ones is really helpful for that. And also to do this in a modular and open way, so that if you want to integrate with other types of analyses, more advanced analyses that we're developing later on, analyses that you develop, you want to throw some QPath segmentation into the middle, that you can integrate and do that too. And so I'm just going to show a couple quick screenshots here to orient you to the type of simple web application that I'm talking about, being able to list and search for things, and of course also being able to look at the data itself. And so for those of you who are software-minded, I did want to include a slide about the architecture of this. This is a very standard, simple architecture we have. The point here is not that we're reinventing this. The point is that we're applying a lot of software best practices and standards to biological technology development. On the analysis and management side. And so just we have a browser-based front end, happens to be using the React library. That's where you as a person go and click buttons and move sliders around. And then again, also allows you, if you're an analyst, a sort of coder, to interact with it and script all of these interactions as well. And then the rest, again, is just having both, some structured data storage as well as some object storage for the images themselves, and having some scalable workers for separate tasks. And that lets us add more intensive image analysis pipelines as well as we expand. And so, when we talk about data management, what does this mean? So one of the questions we ask there is, when I have an image, what is all the information that I want to know as an analyst, as a pathologist, as anyone, for every image that I ever look at? And you can make a laundry list, this is not meant to be complete, of what you're interested in having at your fingertips. That would definitely include the antibody panel, what slide, where on the slide, the type of tissue, all the acquisition settings. But then one of the challenges, it's easy to say, okay, we want all of these things, but you're not going to write down all of those things every time you go and take a picture. That's very repetitive and error prone, and no one would do it, and it's ridiculous. And so, so the other question to think about is, where does this data get created? Where does this metadata get created in the user's workflow? And in this case, it's not super linear. So you may have two different antibody staining panels, and on a single slide, you may stain one region of the slide with one and another region with another. You may have a tissue microarray, like Liat was just talking about. In her case, it was one tissue type, and ours, in another, it might be many tissue types. And so suddenly, your list of images from a single experiment has all of these interconnected lines, matching these things up with each other. And so your database schema ends up looking something like this. This is a real one, but that's okay, because you can have that kind of in the background, and you can write your queries that then let the user see what you see on the left there, just the things they're interested in, but have all this structure to these relationships in the background. And I'm not going to go through all the different tables in there, but just kind of talking specifically about antibody staining panels, which again is obviously not specific to the MIBI. Many of you have talked about other technologies today that use that. And thinking about, especially as things get more multiplexed, higher and higher dimensions, how do you make this convenient? How do you capture all that important stuff? You're doing a titration, you want to not have to repeat delineating all of the different antibodies you use. You want to be able to say, okay, I made it once, now I'm going to dilute, et cetera, et cetera. And this might be done in phases, it might be done by different people. And as came up earlier today with a question, I believe, it's not just what protein is it, you also care about the clone, you probably care about the lot of that, et cetera, et cetera, et cetera. And so, just a kind of applying this general principle of enter these things once, have some master list of all of the antibodies you're using, and then selecting them rather than entering them as you're creating a panel. And the same goes for slides and tissues and all of that, and just having that approach for how we annotate the data to begin with has made things a lot less error prone. And I'm very pleased to say that, unlike in multiple previous jobs of mine, I've spent zero time at Ionpath editing some files that have HLA-DR with a hyphen and others that don't, so that I can analyze them together. It's a big time saver. And of course, having once you have all that information, you have these complex relationships, but that's okay. We just want to display the things that the user most cares about alongside every single image, and then also have these links out to the more detailed information for the slide or for the panel or for the tissue section. I think we're all spoiled because we get this in a lot of other tools, software tools that we use in everyday life, whether it's our address book or a Facebook or whatever. And again, just at a smaller scale, applying that during the process of technology development, I think is very powerful. Quick aside, what you're seeing there is a secondary electron image of the tissue section, because there was a question at the end of Liat's talk about how to choose the field of view, and that's an image that you get in almost real time. I mean, there's maybe less than a second delay of refresh from the top to the bottom of the image that rasters over it, but that you can see and help choose your fields of view as well at the MIBI when talking about field of view selection. Another thing is, again, we want our data to be structured. We want to have these constraints about how that for all these connections, but we also want to be flexible, and we want to say that, okay, there's these antibodies in your panel, and that's what we've imaged, but maybe you're doing analyses that have other sort of derived layers. In this case, maybe I'm showing the cell segmentation boundaries that maybe came from an external tool. Ionpath was mentioned earlier. It's one that we've used. We've also used neural nets for it, and before something's fully integrated with this tool, to be able to just hook it up, put it in there, and then again, I can send to a pathologist, and our pathologist has already been looking at this data, and as I update it, she can say it's getting better, it's getting worse, etc. Maybe not for the segmentation, I can hopefully tell that myself, but often for other methods, I need expert help. I'm going to, another thing that, again, just other useful snapshots, just to, the kinds of plots maybe aren't hard to make, but if you have to make them from scratch each time to do your sanity checks, to do your QC, it can be cumbersome, and just trying to provide them as a tool. I mentioned searching earlier. That was one of the initial main motivations. In this case, what we really wanted was both to have any type of user be able to search any image they've ever acquired and have access to, and then long term, as people make images public, and Liat's already done that with the data that she talked about, and we're very much looking forward to seeing more and more published data sets or other data sets made public, that you can go back and mix and match and find all of the images of interest. And we really think, long term, that's going to be very, very useful for doing these meta-analyses and enabling us to use things that maybe were acquired for one purpose and repurpose them over and over and over again for other questions. And there you also see that you can do the same search through the API, which is, that's often how the software team uses it. The immunology team uses it for the UI. It's the same thing in both. And just as a toy example of the kind of thing you can do with this, more of as a method development example rather than as a science example, is you've developed some segmentation tool. You've used a variety of cytoplasmic or membrane markers as part of it. And now you want to see, do I really need all of them? Or if I replace one with another, is it better or worse? And of course, we want to, you want to use, we have the privilege of having 40 channels available to us, but of course we want to pack as many of them, as many other antibodies in there as we can. And so just being able to go back and say, give me all of the images I've ever had with beta tubulin and HLA class 1, and I'm gonna, just run my prediction over them and see how they compare. Being able to do that in 30 lines of code is rather than having to wait and acquire new data or ask around and hope that what you get matches up has been really a efficiency multiplier for us. Again, I mentioned at the beginning just having standardized open file formats. It seems like a no-brainer if you've been working with data types that have existed for a while. You work with flow cytometry or cytop data, you're like, of course, it's an FCS file. But as a lot of these new things come out, there is that initial step of saying, how do we represent this data? How do we share this data? How do we make it so that things are compatible? And there's been a lot of efforts for this on the imaging front, and we've contributed to them. And so I just wanted to note that here. Let's switch gears a little bit and talk about some visualization, because as much as we love all of the data management stuff for this tool, a big part of it is having this information jointly with being able to see the images themselves. And I think that one thing that can get swept under the rug a little bit is that for some of these mass spec or other imaging technologies that aren't just taking a picture of something illuminated, like a slide under a microscope, there's not necessarily a single canonical visualization, even though we consider it imaging. And And in the spirit of Adib earlier talking about for the scTECH exactly the transformations of the numbers that get it to look like scitop data or other things, going back to quantitatively, what do these numbers represent? How do we rescale them? How do we set axes on them so that they're meaningful? That's similar to what I'm going to reference here. And so just an approximate analogy is that the dynamic range of your camera is actually bigger than what you can display in a picture. And so we're spoiled. It's bigger than what a screen naturally displays is what I meant to say. And so we're all a bit spoiled by this now, only recently, because you go into HDR mode and you see all that nice contrast and the dark stuff and all that nice contrast and the bright stuff without it either being too dark or oversaturated. But that comes from this mapping of what is the light intensity that my camera recorded and remapping that onto your digital display. And that looks like that. And so just as a kind of bringing it back to biological data, and what a lot of you are used to from looking at cytometry data is, maybe a histogram of intensity. It could be intensity per cell. If it's an image, maybe it's intensity per pixel. I'm looking at that, with after some transformation and with the kind of range you set having a huge impact on it. And so in this case, it looks like, oh, that lower plot of CVA, it looks like there's nothing on there. But of course, if I just zoom in a bit, there's actually a big negative and a small positive population. It's just on a different scale than my keratin. And that's fine. We can tailor our scale to different markers, but we don't want to tailor too much because, if we're not looking at other samples, we want to hold our transformations and our scales constant so that we do apples to apples comparisons. And just it's the same with imaging data, whether it's optical imaging or mass spec imaging, where you want to have that flexibility of setting the transformation. Here I'm just changing, the boundaries, but I could have been putting it on a log scale, for example. That flexibility of setting the transformation and the scale that is right for your data set, but also being able to hold that constant and do quantitative or even visually consistent comparisons across data sets. And just, one more example here, if I just have some image, this is just some PNG on my desktop, and I open it with preview on my laptop, and I see black and a couple little white spots, and I'm like, there's no signal. And then just changing that range, there's actually a lot of signal, there's a lot of detail there. And this is something that I've seen, it sounds obvious, but I've seen a lot of people new to other forms of imaging, get surprised by and have to adjust. And it really is, it's a fundamentally different thing than an image like this, which has this canonical visualization, and you don't need to, you have no expectation of having to adjust and, make one thing brighter or change the scale on it. And just from on a quick technical note, in terms of doing some of this in a web application, which has all these nice properties of a centralized data storage and not having to install anything on your computer and all this image sharing stuff, the flip side of that is pulling up an image like this from some data storage is you just have some little image element on your front end, whereas all of a sudden if you're wanting to do these transformations or these manipulations and have it be really responsive for the user, all of a sudden you have to do that in the browser itself, and that's actually been pretty fun, and it's something that I think a few years ago would have been a lot harder to do than it is now. You can do it now with WebGL, which is a GPU accelerated stuff, and someone's talking about that later, and so I won't go into it, but I was excited to see that this use of it is spreading throughout representing biological data in the browser. Anyway, so just kind of some examples of these simple decisions that have a really big effect on how you see your data. Here's one tonsil field of view, and here I'm showing the full range of the double-stranded DNA from its min to its max on this field of view, and I've zoomed in real close to the x-axis of the FOXP3 because I really want to highlight all of those nuclei that are positive. And then I want to hold that constant as I move to another image. But this image might have a bigger underlying scale, and so I want to be able to expand it out to that one. And then maybe I lose a little contrast in the FOXP3 if I want to see the DNA more. And so again, it depends what you're looking at. It depends whether you're that there's not in my opinion, there's not a single correct setting. Once you get into these higher dimensional stuff where you want to be able to highlight different channels and switch between them real fast. And so just making that interactive and intuitive for these QC checks, for you're giving data to a pathologist who's never worked with this particular technology before, just making it quick for the interaction. Of course, a lot of this is motivated by the fact that we are looking at more than just 1 or 2 channels. And so one of the things one of the reasons we use this a lot is, of course, the visual ability to look at co-localization. And here I've just shown some examples of that with some immune markers where we have a really strong expectation about where they should and shouldn't be co-expressed, and we see this beautiful consistency with CD68 and CD3 only being present on the CD45 positive cells. The same with my T cell subsets, separation between the CD45 and keratin. And of course, this gives you confidence that when you go and you see co-expression in markers where you don't have as much prior expectations about where it should and shouldn't be, you can pick up those same things. For example, in this particular case, this is the same image as the previous slide, we saw PD-L1 only on macrophages. We saw PD-1 split between the CD4 T cells and the CD8 T cells. And then we were able to, by having this quick browsing in a really straightforward way, to then it kind of triggers these downstream quantitative analyses of saying, okay, that you can then compare across a larger cohort of images in terms of, is PD-L1 expressed on my macrophages more in these samples than others, and what does that correlate with? I'm running out of time, so I'm just gonna real quick show that you can look at lots of colors at the same time. Of course, it's diminishing returns at a certain point. And if you really are interested in PD-L1 versus CD3 and where they show up, you probably want to look at this view instead. And I'm gonna, I think, in the interest of time, skip some of my logic on exactly how and why we combine them in certain ways. But what I will point out is that again, just connecting what you're looking at quantitatively to the underlying data and thinking about, as you're combining more than 3 colors, you are again doing a mathematical transformation once you have more than 3 on the underlying data, because your RGB color space is 3D. And so just being aware again of what you're seeing is a representation of some ion counts, and especially when you're looking at validation and such, to go back and think about, how does this relate to what I actually measured? And the last point I'll make is just about how a lot of the motivation again was about sharing of data once something is complete, but even more so of that, even way before that, just being able to use rapid iterations between the computational person and the scientist and the physician, and so that everyone's looking at the same data, and so that you can integrate as you have methods under development, and so on and so forth, and to do so in a easy and flexible way. And with that, I'd just like to summarize and say that, of course, there's some extra effort at the very beginning of a project or a technology development if you choose to put some of the structure in place. But we found it to be well worth it, because now, as we're getting to the stage where we're asking more scientific questions with the data. We have collaborators and ourselves working on more analysis methods. We have this foundation on which to build. And it's easier than it's ever been. Again, we're not inventing new software methods here. We're taking advantage of all the stuff at our disposal. And so I'd like to thank some of the computational folks at Ionpath and the entire Ionpath team for their work on this as well.
We have time for one question.
So right now, this piece of software is available for adding data to it for all MIDI users, and we have a public instance of it for other members of the public, such as yourself, to look at and download the data that's available. And so if you're a MIB user, you should have access, and if you're interested in using it someday with other data types, I'd be happy to chat about that.